Please do come back at 9 o'clock. I am going to go through GCSE though right now. Okay, so one of the key things that is always interesting when it comes to understanding anything to do with organisation is that we have to take a step back and remind ourselves about cells. So cells are eukaryotic if they have nucleuses that contain, so nuclei plural, nucleus singular. So if a nucleus contains genetic material like the chromosomes, we know that that therefore is a eukaryotic organism. So we look at the nucleus, we look at the cytoplasm, which is the site of chemical reaction. We know that we've got our cell membrane, this barrier. We know we've got our ribosomes. We know that we have got that site for protein synthesis, the ribosome. We know we've got a site of aerobic respiration with our mitochondrion, or mitochondria, plural or singular. Make sure you're looking at what they're labeling. And we have, if we're looking at plant cells, chloroplast, which I talked about in last week's live, talking about how photosynthesis occurs. So we also have the cell wall, and the cell wall obviously is going to be strengthened so that it can support the cell in the plant cells. And we talked about how that helps with osmosis 2 weeks ago. And the other thing to note when we're looking at plant cells is the permanent vacuole. So this is where we've got the cell sap, which is keeping the cells rigid so that they, again, have this support. Now, why are we interested in taking a step back and looking at our cells is because if you understand the different parts of the cell, so this is GCSE level, if you understand the different parts of the cell, what you then need to understand is, well, how is it that it goes from single-celled organisms up to being multicellular organisms like us, which is where I want to take you today. So we have got these cells, and we know that these cells, animals and plant cells, are bigger than prokaryotic cells because the size of a prokaryotic cell is very similar to the size of a mitochondrion and the size of a chloroplast. So one of the ways they may assess your knowledge in your GCSE is seeing if you understand how the size of something can determine whether we're looking at that simplified, still quite complex, but prokaryotic cells, bacterial cells, or if we're looking at eukaryotic cells that have got more things inside, like the mitochondria and the chloroplast. So what I'm really interested in is taking you on a journey. So if we understand that our nucleus that we just talked about contains our DNA, we should know that when we talk about this cell going through mitotic division, if we're going to end up with two daughter cells, what we have to understand is our DNA needs to be replicated. So that's the first stage in the cell cycle. Then after the DNA has been replicated, we also need to increase those organelles we just talked about. The mitochondria, the ribosome, all the other organelles need to increase in number so that the cell, when it goes through that division, has enough so that both cells have the same organelles. Then we go to stage 2. Now remember, this is GCSE level, so if you are an A-level student who is popping in, then please do come back at 9 o'clock. Right now, I'm just covering the GCSE knowledge for mitosis and moving on to organisation. So the DNA has been replicated. Now what we need to do is we need for one set of each of these chromosomes to be pulled apart. And they're going to be pulled to the poles of the cell. Now, if you're going on to do A level, you'll learn so much more about how those chromosomes are pulled apart, about how that replication occurs. But for GCSE level, this is your stepping board to get you ready for that next stage. So the key thing about the second stage is that nucleus is going to divide. So for your GCSE level, you are saying your DNA is replicated, stage 1, and the organelles. Stage 2, the nucleus is going to divide. Stage 3, This is where the cytoplasm and the cell membrane is going to divide, forming two identical daughter cells. Guys, if you are joining me here on TikTok or on YouTube, please do, if you have questions as I'm going, please do ask the questions. I am live right now. I am available to answer any of your questions, but I'll just keep teaching you because this means it's there for you to look back and get ready for those exams that you will have in May and June. Okay, so why did I want to talk about this? I want to talk about this because you need to understand for organization that your cells are these basic building blocks of all living organisms. You need to understand that they will go through mitotic division, mitosis will occur, so that we can get daughter cells. We can get cells that are genetically identical to the cell that we started off with. So when you first were produced, in a sense, when sexual reproduction occurred and the egg and the sperm came together, that fusion of the egg and the sperm produced one fertilized egg cell, a zygote. That zygote then goes through many mitotic divisions to produce you, this multicellular organism. Now, as your cells differentiate, what then happens is these cells switch on or off the genes depending on what's needed to specialize into the role that they have to play. So a tissue is a group of cells with a similar structure and function. So you need to know potentially about muscular tissue, the contraction that happens in the muscles, the glandular tissue, the secretion of enzymes and hormones. And we're going to focus on that. Today's lesson is about those glands and how and where these enzymes are secreted. And you also have your epithelial tissue, which covers the outside of the body, covers the outside of these internal organs. So we have the cells, a group of cells making a tissue. And then if we go to our organs, these are several tissues that are working together and they're going to perform a specific function. So again, similar words that we're talking about here. We had our muscular tissue, which was contracting. So when we're in the stomach, the muscular tissue is needed to churn the food up. So our glandular tissue in the stomach is going to be producing our digestive enzymes, which are our pepsin. Our epithelial tissue is the one that's going to cover the inside and the outside of the stomach. So we have this organization of the cells inside our body to allow us to do the jobs that we need to do. And in this case, whilst we are looking at the digestive system, we're going to look at the fact that there are different organs working together in a system, the digestive system, and that allows us to digest and also absorb the food. So what are our organs? Well, there are certain ones that you need to be able to label. So if we start with our esophagus. So the esophagus is the tube that runs all the way down, and the esophagus is actually really important because you know about the tissues, the muscular tissues. As you are eating food, you should be chopping it up with your teeth, getting it down into smaller bite sizes so that you are increasing the surface area for enzymes to be able to work at breaking down the molecules that are within the food that you've eaten. And if you've ever not chewed enough, what you may find, and I've done this on several occasions, is you actually feel like this bolus of food gets stuck in your throat and you can feel it slowly making its way down through your esophagus and you're like, oh, I can feel every movement that's going all the way down. And that's because what you have is you have these muscles that are going to squeeze, they're going to contract, and they're going to squeeze and push the bolus of food all the way down your esophagus. So you have different muscles, and those muscles, as they are contracting, cause what's known as peristalsis and the movement of the food down in through your esophagus into your stomach. So once we've come all the way down our esophagus, what we'll then do is our food will enter our stomach. Now, our stomach is a really interesting organ because our stomach has got the stomach acid that you should know, which is hydrochloric acid. So your stomach acid is actually going to contain hydrochloric acid. And so many students get this wrong. They think the hydrochloric acid is there for breaking the food down, but the hydrochloric acid isn't. The hydrochloric acid is there as a protective mechanism in case, unfortunately, you've taken in some food that's carrying some microorganisms. So this acidic condition is a way in helping you actually protect the body against any type of pathogen that might have been swallowed with your food. It doesn't always work, however. Sometimes you obviously have taken in some food and you will have a reflux where the body will actually cause you to be sick so that you can expel the pathogen. With the vomiting syndrome that comes, gotta love it, but not at the same time. I hate when I get hit with the vomiting bug, and that's because I get hit bad with it, and every time you will feel as the stomach acid comes up through your esophagus and out through your mouth, you get this burning sensation. That is the acid. Now this is a stomach acid, hydrochloric acid, which has a low pH. That is going to actually cause harm if it happens too many times. So being sick is not a nice experience for anyone, but it is a protective mechanism that your body does. Right, moving on then with the digestive system. We then move from the stomach to start with our small intestine. So we've got our small intestine, then it moves into our large intestine, and then it comes down to the bottom area here, which is our rectum, and then our anus. And we ingest food, we take food in, we egest it out. So if you think about all the way from your mouth, down your esophagus, your stomach, your small intestines, large intestines, all the way down, it's just a really long tube. And so what you have is you take it in, and anything that you don't actually absorb after you have digested comes out the body. That is you egesting. You are releasing it out of the body because it is waste material. And that's really important when you get to things like how the microorganisms are able to actually recycle nutrients back into the ecosystem, because what's left in the feces that we egest, that is going to be broken down by any of those microorganisms. So biology is amazing if you can think of it like the bigger picture rather than just this topic area, that topic area. All of it is interlinked. So let's not forget, we're talking about the digestive system, we're talking about organs, several organs working together so that they can carry out that function of digestion and absorption of the food. So we also have to talk about the liver, because the liver is really important in producing the bile, and that will get stored in our gallbladder. So you should, for GCSE level, be able to identify all the different organs within your digestive system. But not only do you have to be able to label them, you also have to talk about what happens at the different organs. So we can't forget about one of the really important ones, the pancreas. So the pancreas is a really important organ because what it's going to do is it is going to produce some of these enzymes that we're going to talk about that are needed for that digestion process. So let's start with our enzymes and let's go back up to our mouth. Let's talk about what's happening here. So last week we talked, I say we, you were all here, obviously, or if you weren't, please do check out my video from last week. When we talk about enzymes, and we talk about enzymes having the right active site, and the active site which is complementary to the substrate that it's going to work on. For GCSE, you need to know specific named examples. And if you're an A-level student who has stumbled upon this, AQA A-level, you need to know this in a little bit more detail. But let's just start with the GCSE side of things. We have got salivary glands inside our mouth, and I already spoke about the fact your teeth are there to break your food down into smaller chunks, increasing the surface area for enzymes to work on. Now, your salivary glands will also produce a enzyme called amylase, and this is one of the hints that I always say to all of my students. The minute you see A's at the end, that is telling you that that is an enzyme. So amylase is also known as a carbohydrate. So GCSE level, carbohydrates breaks down carbohydrates. So if I'm looking specifically at amylase, amylase is a specific example of a carbohydrate. So if you can't remember amylase, just say carbohydrates. If they're asking about what breaks down carbohydrates, it is carbohydrates. So it's going to break down starch. Now, we're starting with it being produced in our salivary glands. So we're going to start that digestion there in the mouth. So my amylase and my starch are going to collide. When they collide, they are going to fit together quite nicely. Remember GCSE lock and key model. They're going to fit together quite nicely because they are complementary. Try to get into habit of using that word. That will result in the breaking down of the starch, the really big polymer, into smaller carbohydrates. What's then going to happen is you will also produce other amylases, pancreatic amylase, and that is going to flow from the pancreas all the way down and enter the small intestine. So you can see in my picture here that I have got a tube that leads down from the pancreas right the way down. It's a pancreatic duct that leads into the small intestine. I'm not actually doing it specifically for any exam board. However, I teach mainly AQA. So I would say this is AQA, but can actually, because it's GCSE, cover pretty much majority of the exam boards. It's just about how they ask the question H. But thank you for asking your question. So the key thing to note is the pancreas is going to produce our enzymes, and they're going to be released into the small intestine, and that is where most of that starch is digested to the simple sugars. Now, I'm going to stick with AQA at the moment. AQA have required practical number 4, where you have to investigate the effect of the pH on the rate of reaction of amylase enzymes. So most of the different exam boards will have their own required practical, but pretty much, because if it's AQA at A level, you go through the same stuff. It's pretty much the same for most exam boards, because this is one of our favorite ones to use. If it's not amylase, it will be trypsin. But right now I'm going to talk about amylase, and then I'll bring in trypsin a bit later on. So I'm going to try and give a broad spectrum on these sessions so that if you are different exam boards, it's still relevant to you. So we are looking at the effect of pH on the rate of a reaction. So what I would do is I would prepare an amylase solution at a pH of 5. So this is where you need to bring in your skills of what is it I'm talking about here. Is it my independent variable? Is it my dependent variable? And what are my control variables? So anytime you do any type of practical, or you're asked any questions about a practical, your head should straight away go, right, what's my independent variable? Well, they've already said this here. Investigate the effect of pH. So my independent variable will be that I'm going to change the pH that the amylase is going to be exposed to. So if I'm going to be changing the pH, what do I need to control? I need to control things like the concentration of a solution. I need to control the volume of the solution. So it's really important that you have a set of criteria that you can go to, and then you adapt to the question that they've asked you in the exam. So if it's the required practical they want to assess, it will be about pH. If it's just about enzymes in general, it will be potentially about any of the others. I want to control, for example, my temperature. So I've prepared an amylase solution at a pH of 5. So I'm going to control the concentration of amylase. I'm going to control the volume of my amylase solution. I'm going to control the temperature at which the enzyme is exposed to. I'm going to control the fact that what I'm looking at with this amylase solution is I'm going to look at its rate of reaction of it. Well, what does amylase break down? We know that amylase breaks down starch. So therefore, I need to mix that amylase solution with starch. So I need to control the concentration of my starch solution. I need to control the volume of my starch solution. So those labels that you have, if you have a set criteria, my concentration, my mass, my volume, my temperature, my pH, I'm changing my pH, so therefore I need to keep all the other things the same. They will ask you and expect you to have about 3 of them, okay? So do make sure you have a list, but make sure you relate it back to whatever the question is that they are asking in that exam question. So in this case, I'm going to be looking at the rate of the reaction, my amylase. So my amylase breaking down the starch. Now, again, going back and double checking, if I remember, because we're talking about starch here, I should know what happens when I do my food test. So if I have a starch solution and I put it into a spotting tile and I have some iodine solution in there, what is going to happen? Well, I should know that if I remove a drop of the amylase and starch mixture every 30 seconds and test it for the presence of starch, if starch is present, it will go a blue-black color. What are we looking at here? We're looking at the fact we're going to remove a drop, we're removing it every 30 seconds, we're testing for the presence of starch, and we're going to keep going until the starch has been digested. So it could be about 10 minutes. Now, if it's up to 10 minutes, I'm going to have to keep going. So I may have to use 2 spotting tiles, as you can see here, and I would record how long it takes until the iodine solution stays the negative result that I would expect. So this is where they could be testing your knowledge on, do you know that if it's blue-black, it is a positive result? And if it has stayed orange-brown, that is showing a negative result, and therefore all the starch would have to be digested. There's no more substrate left, so that all the starch has been digested. If it continued to be blue-black, that would mean that there's still starch present, so not all of it has been digested. So this is where they're expecting you to apply your knowledge to whatever question that they give you. Now, it could be that they give it to you in a graph format. So if they're giving it to you graph format, what have we got? Well, the time taken to digest all the starch in minutes, we can see this coming down here, and here's my pH. So what's happened then is I can see that the time, it gets less time, less time, less time, less time, less time, and then more time again, more time, more time. So what does that mean? Well, it took a long time to digest the starch. It didn't take as long, and it took a long time again. So this is what it would look like if you were actually doing the practical. This is what it looks like when you're doing it and seeing it in a graph format. So it really depends what scale they're going to assess you on when it comes to how they present the data and what they want you to give them. So you should know when you are looking at the graph or whether you're looking at a table of results, you should be able to identify where you think that optimum pH is. So if it is that they have given you as a graph, you should know that the point at which it had the least amount of time, that is the optimum pH. But am I sure on that? How many repeats did they do? So this is something you need to think about. How many repeats did they actually do? Did they do it enough times to give you a valid set of results? Or should they repeat? And should they repeat between a smaller number of intervals between pHs 6 to 8? So if I'm going to repeat this, I would probably want to repeat between 6 to 8 so that I could be sure that my optimum pH was actually 6.8. So yeah, guys, if you just joined me on YouTube, supposedly I've been talking and nobody's been able to hear me, which is quite funny. Okay, so I'm going to keep going anyway, and hopefully that will have fixed it. I have no idea why that wasn't working there. Okay, so what I was saying before is when you're talking about the pH is the optimum. I'm saying by looking at the graph, the optimum is about 6.8, but I would want to repeat this, and I'd want to repeat this between 6 to 8, the pH of 6 to 8, probably doing maybe 6.2, 6.4, 6.6, etc. I would do smaller increments between 6 to 8 so I could be sure of what I have suggested as being my optimum pH. Guys, if anyone is still watching on YouTube and you can hear me now, I'd really appreciate if you could answer or write a little message to say that you can actually hear me in there. That would be really great. Okay, so let's move on and put this down with regards to, we've looked at amylase, we've looked at what happens with amylase. Now we're going to look at proteins and where are the enzymes produced to break down our proteins. Our foods, the nutrients that we take in, should have proteins in them. They should have, oh, somebody messaged, no. Guys, if anyone is on YouTube and can hear me, please do make a comment so I can see that you can definitely hear me. It's not A-level. A-level will be back at 9 o'clock. So if you're looking for A-level, please come to the 9 o'clock one. This is the GCSE foundational, I say foundation, higher level, but the foundational level ready for A-level. So I'm trying to help out GCSE students and those A-level students who miss those foundational skills needed for the A-level. So it is helpful, but I'm not going to go to A-level content yet. I'll be back at 9 for that. Right, what have we got? Well, we know that we've got a protease, and a protease is going to break down proteins into amino acids. So we know that our proteins are polymers. We know that the proteins are broken down into the amino acids. And so we need to know, where do these proteases come from? So the proteases are actually produced first in your stomach, and then you also have them produced again in your pancreas and also in your small intestine. Now, we spoke about the effect of pH on amylase earlier, and so therefore we now need to bring in what happens to the enzyme when we change the pH away from the optimum. So pepsin was the one which was produced in the stomach. Now the stomach, we've already talked about, has got hydrochloric acid in it. So therefore, when we are digesting our food, when we've taken the food in, when the food is in our stomach, for us to start the digestion of the proteins in our stomach, we are actually down to a pH of 2. So the optimum pH for pepsin is 2, and that's why pepsin is the one that is located in our stomach. So we have glands in our stomach which is going to produce and secrete the pepsin into the stomach, and then the pepsin is going to be able to break down the proteins into smaller peptides and then into those amino acids. Now, those hydrogen ions actually interact with the enzyme structure. So if we have a pH that goes away from the optimum pH, we actually denature the enzyme. So the further away we go from the optimum, the active site starts to change its shape. And because it starts to change its shape, it is no longer able to function at the same rate as it was before. So when we look at enzymes, we should know that there are different enzymes, and different enzymes have got different optimum pHs. So you have to be sure what you're looking at is in the question what they are telling. Don't memorize these pHs. Make sure what you're actually doing is you're looking at the graph that they've given you or the data that they've given you, and you are then looking at where is the fastest rate of the reaction happening, and that fastest rate is showing you where that optimum is. So we know that in our stomach we have got our acidic conditions, and we know that the pancreas and the small intestine produce different enzymes to help with the digestive processes that occur. So the next enzyme we need to look at is the enzyme that's going to break down the fats and the oils, the lipids, which are in the foods that we've eaten. So we know that because we're talking about an enzyme, it's got an A's at the end. We're therefore talking about lipase. So the lipids are broken down by lipase. Now we should know, whether we're A level or whether we're GCSE, we should know that our lipids contain, GCSE level, glycerol and 3 fatty acids. That's known as a triglyceride. So a triglyceride, our lipids, is going to be broken down into those monomers, which are our 3 fatty acids and our glycerol molecule. So if we understand that the enzyme lipase is going to break down those lipids, and we understand that it's made in the pancreas and the small intestine, we know that it's going to help digest those lipids. However, fats are really funny molecules. So when we take in our fats, and I've got to keep myself at GCSE level here, the fats actually clump together in big clumps. So we know that our stomach was acidic. We know that our stomach gets emptied into our small intestine. So therefore, the contents of the stomach actually have got a pH of 2. When they get emptied into the small intestine, we need a way of neutralizing the hydrochloric acid. So this is where your chemistry knowledge should be coming in. You should know about your pH. You should know that your pH goes from all the way from 0 up to 7, then up to 14. So if I'm looking at something that is acidic, I'm going to be making my way down towards 0. If I'm looking at something that is alkaline, I'm going up towards 14. When I'm in the middle, if I'm neutralizing, I have got a pH of 7. Now, for those of you who are obviously doing your chemistry as well and are considering going on, you'll be learning about your hydrogen ions. So the pH is the potential for hydrogen ions. It's about whether the dissociation of those hydrogen ions, does it fully ionize to release hydrogen ions? And when we look at the alkaline, we're talking about hydroxide ions. So if we have hydrogen ions and if we have hydroxide ions and those two come together, they actually produce water, H2O. And that's why we get a neutral solution. So if we are looking at the bile that's made in the liver, that's stored in the gallbladder, that bile is actually an alkaline molecule which is going to neutralize the hydrochloric acid. So we're going to put a alkaline solution into the small intestine. We have an acidic solution coming into the small intestine from the stomach. So the two of them come together is going to cause neutralization to occur. We are neutralizing the acid from the stomach. But what we also do is that bile is going to emulsify, take this big massive fat droplet and break it down into smaller droplets. So that emulsification that's happening, taking it from being a large molecule with a small surface area to these smaller molecules with an increased surface area, it means that the lipase can actually work faster because it can access the lipids quicker than it could when it was one big fat droplet. So if you were actually looking at the fat droplet and you were looking at the way in which the glycerol molecules lie, they actually lie with their little tails, those fatty acids, all in the inside. Well, we should know that the lipase needs to cut those fatty acids and release them. And because it needs to cut them and release them, it therefore means that it needs access to them. And that's where the emulsification comes in, because the emulsification is about taking it from a big fat droplet down into smaller droplets. And then the last little bit that I will finish on today is about homeostasis and regulation of internal conditions. So we know we've got our cells. We know we're talking potentially about a whole organism. Homeostasis is this regulation of the internal conditions of the cell or of the organism. And we need it to maintain these optimum conditions. So we know we've got our enzymes that have their optimum pH, that have their optimum temperature. We need to understand that there is also conditions like blood glucose concentration that needs to be maintained, which is the optimum. We need to understand that body temperature, and we need to understand our water level. So I'll go into that in more detail next week, but just to touch on it quickly, blood glucose concentration, we talked about this in a couple of weeks ago, the mitochondria, the site of aerobic respiration. Glucose is required for respiration, whether it's aerobic in the mitochondria or whether it's anaerobic, it needs it to be present. How does emulsification increase the surface area? Okay, so emulsification increase, let me go back a second because that's a good question. Sorry, I missed it there. Let me just put in an extra one. Here we go. So if I go from having one big cube, okay, so if I've got one big cube and you think about the surface area would be 10 by 10, and then there's 1, 2, 3, 4, 5, 6 sides. So if we're looking at my surface area, we would do 6 times 10 times 10. Okay, so the surface area would be 600, wouldn't it? So if I was then to look at how many cubes could I get in there? I could get 10 cubes, all one by one, each with 6. So I'm just going to go from the bottom row, but obviously you'd have the same going up, 10 by 10 and so forth. So if I take 1 cube and I've got 1 by 1, I've got 6 times 1 times 1. So if I've now got that times 10 going up times 10 going across, that therefore is going to increase the surface area that's exposed. So when you look at surface area, you're looking at all the points at which the enzyme has access to. So all the points at which the enzyme has access to is the surface area. So if I have got all these little cubes inside and they're no longer touching each other. So what I mean by that is if I had two cubes side by side, my surface area only hits there. Whereas when I have the two cubes being separated, my surface area has now increased. Does that make sense? I hope that makes sense. Guys, I am going to finish now because I have to set myself up for my group tuition and